going to talk about tissues. In the body we have four main categories of tissues. Nervous tissue, which is going to be responsible for our internal communication. And nervous tissue can be found in the organs, your brain, your spinal cord, and your nerves. We're going to talk a little bit about the cells in nervous tissue today, but we'll talk about nervous tissue in detail when we get to the nervous system at the end of the semester. The other type of tissue, the next type of tissue that we have is muscle tissue. Muscle tissue is contractile, so it causes some kind of movement. It either moves your bones so that you can make movements, or it moves stuff through stuff. So skeletal muscle moves bones, cardiac muscle moves your heart and pumps blood, and then all of your hollow organs have layers of smooth muscle, and that's going to push stuff through your hollow organs. What we're going to spend most of today talking about are epithelial and connective tissues. Epithelial tissues form boundaries between different environments in you and between you and the external environment. Anything that goes into or out of your body does so through an epithelial tissue. So examples that are showing you here, the surface of your skin. Your skin is multi-layered, it's got connective tissue underneath it, but the first surface on top, your epidermis, is epithelial tissue. And then the lining of all of your digestive tract organs is epithelial tissue. And most of your other hollow organs as well. I guess I can't really think of an exception. And then connective tissues are the last broad category, and they support, protect, and bind other tissues together. Examples would be like bones. It's the only solid tissue in your body. Tendons, which hold our muscles to our bones. Fat is a connective tissue. Blood's a connective tissue. It's the only liquid tissue in your body. So we'll talk about all of these in detail today. So we'll start with epithelial tissues. We're going to talk about epithelial tissues first, then we'll take our break. Then I'll give you a couple minutes to work on this worksheet I brought, and we'll talk about connective tissues, then we'll have our lunch breaks, then we'll do our lab. In lab today, I've dedicated lab today and next Friday to tissues, so you really only have to go through half of the tissues, but you can go through them all if you want. I'll have the TA put them all out so that we can look at them, because we're going to be talking about all our epithelial and connective tissues today. So if you just want to see them, then that would be good. So as far as functions of epithelial tissues go, they are going to be the first boundary between us and everything else. So they help with protection. Is this too light to see at the back of the room? Can you see this blue? Okay. So that's the first function, and they protect us by forming boundaries. Another thing that epithelial tissues do is they secrete stuff. So a lot of our glands in our skin especially are going to make things like sweat and secrete it. So secretion is to produce and release a product. And we'll talk about exocrine glands generally today. We'll talk about specific ones when we talk about the skin. So the secretion is going to be to produce and release a product. Another thing that our epithelial tissues do is absorb stuff. So absorption, which is bringing things into the body. They also help with excretion, which is getting waste out of the body. They are the boundary between us and the external environment, so they have to be able to sense when things are changing, so they're also important for sensation. And we can say they contain receptors that detect changes in external variables. One last function, some epithelial tissues do what's called filtration. Filtration is a passive process where we push substances through a semi-permeable membrane. So in your kidneys, they're going to filter your blood and pull out waste. And so epithelial tissues make up the tubules that are doing that. So this would be how we're going to pull things out of the blood. So epithelial tissues, again, are going to help with protection. That's because they're forming all of our boundaries. Our boundary between us and the external environment, and then even on the inside of your body, like when you eat something, before it can get into your body, I mean, you feel like it's in you as soon as you've eaten it, but it hasn't become part of you until you absorb it through an epithelial layer. So that helps with protection, because if there's anything in it, like bacteria or stuff, then we can have immune cells there in the epithelial tissue that can gobble it up. So it's going to form the boundary between you and the external environment. Epithelial tissues are also going to be important for secretion. We'll see some specific secretions that are produced next week when we start talking about the epithelium. But secretion is to produce and release a product like sweat. Absorption is going to be what, like in our GI tract, when you absorb nutrients in the small intestine, they come through an epithelial barrier and then become part of you. For excretion, this is how we're going to remove waste from the body. For sensation, our epithelial tissue contains sensory receptors. These are going to be important for picking up changes in some variable, be it temperature or pH or something like that, and then they'll communicate it back to your nervous system so that your nervous system can do something about it. And then filtration, which is pulling things out of the blood. They can act as a semi-permeable membrane that we can swish stuff out of and through. Or as far as characteristics of epithelial tissues go, all epithelial tissues have some things in common. And these are going to be general characteristics that are shared between all of them. The first characteristic is what we call polarity. Polarity is sidedness. So they're going to have different surfaces. And this is caused by the fact that all of our epithelial tissues are attached to what's called a basement membrane. So a basement membrane is non-living tissue that is secreted by the epithelial tissue and the connective tissue below it. They share this basement membrane, and that's going to give our epithelial tissue size. So all of our epithelial tissues have cells that are going to be attached to the basement membrane. That would be the basal surface. So the basal surface of an epithelial tissue is attached to the basement membrane. Now, we have different types of epithelial tissue. Sometimes they're just one cell layer thick. Sometimes they're multiple cell layers thick. If they're multiple cell layers thick, then all of the cells attached would be the basal surface of the tissue. And then the opposite side that's floating freely is what we call an apical surface. And this faces the free space. So open spaces in tissues are called lumens. In the body, there's no open space. It's full of some kind of fluid. But if you're full of digestive juice or something, then that open space would be the lumen. Your apical surface is going to face the free space, which is the lumen. And so in what this is a simple epithelial tissue with just one layer, then one part of our cell is attached to the basement membrane, that would be the basal surface of the cell, and the apical surface is facing the lumen and is floating free. You can also have tissues that have multiple cell layers. So this would be the basal surface, and this would be the apical surface.
So it's just referring to the location on the cell, basically, the top and the bottom?
It's kind of referring to the location in relation to the basement membrane. So like your skin has 10 to 20 layers of dead cells on top. So this is all the apical surface of the epithelium of your skin. All of the cells that are attached to the basement membrane are still alive. And in your skin, there are several layers between apical and basal. Does that kind of make sense?
Do they have a different name?
The different layers?
Like in between apical and basal?
They do. And we'll talk about that when we go into the integument next week. So yes, absolutely they do. We've got 5 distinct layers in the epidermis that we'll talk about starting next week. Any questions on polarity? Yeah?
So when you talk about skin, would the basement of the skin be the top of the dermis, or?
It's attached to the dermis, but it's still part of the epidermis that's attached to the basement membrane. So in your epidermis, the bottom layer is called your stratum basale. That's where we have living cells that are constantly undergoing mitosis. They're attached to the basement membrane, and then right below that is the dermis. So it's right below, you always have a connective tissue right below the basement membrane. And the reason that that is because epithelial tissues are actually avascular. So I'll go out of order, I think, of your book goes in, because this kind of helps us to understand. So our epithelial tissues, the second characteristic of all epithelial tissues is that they're avascular, which means they have no blood supply of their own. So all of the blood supply, all of the nutrient and gas exchange that's happening to support our epithelial tissues is happening because of the underlying connective tissue. So that's another characteristic of epithelial tissues, is that they are supported by connective tissues. Because they don't have vascularity, but they still are going to need nutrients, they're still going to need oxygen, they're still going to need to exchange carbon dioxide, get rid of their waste. So they do that through the connections that they have with connective tissues. So we'll say that these underlie all epithelial tissues and provide support. We could say through vasculature. So one characteristic is they're all polar. They all have these sides. The second characteristic is they're all avascular, meaning that they don't have their own blood supply. That leads to the third characteristic, which is that they are all supported by connective tissues. So the underlying connective tissue will have the blood vessels that are providing the nutrient and gas exchange for the overlying epithelial tissue. Do you have a question?
And these are true for both simple and stratified?
Yes. Yep. Good question. All right. So another characteristic of our epithelial tissues is that they have what are called specialized contacts. So our basal cells are all going to contact the basement membrane. So that's one specialized contact. But then they also have lateral contacts. So our lateral contacts would be how our cells are linking up side to side. And this can happen through desmosomes, which are like snap-like junctions that are snapping plasma membranes together, or this can happen through tight junctions, which are like sealing up and sewing them together. So that would be another type of specialized contact. Gap junctions aren't found all over the body. They're found in just a couple tissues where we need to have chemical communication that happens instantaneously. So, but we do find desmosomes and tight junctions all over the place. All right. Another characteristic. Are we good so far on characteristics? Any other questions? Another characteristic of all our epithelial tissues is they're highly innervated. This means that they have sensory receptors that are linked to sensory neurons to communicate to the central nervous system about changes. So since that is our first point of contact with the external environment, it's important to be able to communicate changes right away. So we've got lots of sensory receptors that we'll talk about at the end of the semester. These are going to be linked to sensory neurons that communicate with the central nervous system. The last characteristic of all epithelial tissues is that they regenerate very quickly. So they all have regeneration, hold on, as a part of what they do. So how do they regenerate? They always undergo mitosis to give rise to new cells. So we'll say that they're going to undergo mitosis to replace dead or dying cells. And if we think about where we find epithelial tissues, this makes sense. You find epithelial tissues on all your boundaries. So your skin is continually sloughed off as you scratch or you pull on your coat or whatever. You're going to rub off those dead layers. So we've got to be always making new living layers underneath and then killing them off as they rise to the surface so that we can continually replace that tissue. If you think about like the lining of your digestive tract, all of those cells are exposed to digestive enzymes. So they're not going to live very long. So we need to be able to replace those pretty regularly. This is another thing about epithelial tissues, that they are highly mitotic. They always regenerate because they're always being exposed to environmental stressors. Right, so we have two things that we classify our epithelial tissues based on. One, how many layers there are, and two, the cell shape at the apical surface. So the first way that we classify our epithelial tissues is based on how many layers there are. If there's just one layer, we call this a simple tissue. And it's some kind of simple something tissue. So we'll look at where we get our other name in just a minute. If we've got two or more, we call this a stratified tissue. So the first basis of classification is cell layers. If you've just got one layer, you're a simple something tissue. If you've got two or more layers, you're a stratified something tissue. This something is the second part that we use for classification, and that's the cell shape at your apical surface. So if we look at the different cell shapes that are possible at the apical surface, we've got three. Squamous, which are like short and flat. Squamous cells provide the thinnest possible barrier in the body. So you find simple squamous epithelial tissues in places where we need to have like rapid nutrient and gas exchange. So simple squamous cells make up your capillaries. Simple squamous cells make up your alveolar sacs, which are your air sacs in your lungs. So they are thin and they are flat. So they're really good for the rapid diffusion of materials across them. I'm going to rate 4 and 5 now. The next shape that we have is cuboidal. Cuboidal cells are about as wide as they are tall. So they kind of look like cubes. I mean, not really when we look under a cell. They have rounded edges, but they're about as wide as tall. Another thing about cuboidal cells is that they have spherically shaped, centrally located nuclei. So again, they're about as tall as they are wide, and they've got this spherically shaped, centrally located nucleus. Our last type of cell is called columnar, and it kind of looks like a column. And columnar cells are taller than they are wide. And they have an ovoid-shaped, basally located nucleus. So when you start looking at epithelial tissues today and you look at columnar cells, you'll be able to recognize them because they have their nuclei all stained dark. You find them near the basement membrane, and then the rest of them stains pretty lightly relative to the bottom where the nucleus is. So that will tell you where your basal surface is and where your apical surface is. So now we'll look at specifics in the body. So this is our simple squamous epithelium. Again, this is a single cell layer thick. That's what makes it simple. It's squamous because those cells are short and flat. And these are so thin that they are going to allow materials to rapidly diffuse across them. So you find these tissues in places where we really need to be able to do that quickly. So the lungs, I think, are the best example because people really understand breathing and gas exchange. So you need to be able to get oxygen and carbon dioxide back and forth across this simple membrane. So these are the alveoli, or your air sacs of your lungs. And you can see the darker staining spot would be the nucleus of a cell, and then the rest of it's the rest of the cell. So that's one place. Another place we find them is this portion of the kidney called the glomerulus. I think that's what your lab is asking you to look at. But the kidney has simple cuboidal tissue in it. So I don't recommend looking, I mean, you can look for it there, and I can help you find it there, but there's a lot of other tissue there. So if you really want to just see simple squamous, grab a lung slide, and you'll be able to see simple squamous very easily there, because that's all that's going on in our air sacs. So other places, the endocardium of your heart, that's the lining of your heart, the lining of your blood vessels, the lining of your lymphatic vessels, and the lining of those, of all of your serosa membranes are all made of simple squamous epithelial tissue. And again, it's going to help to allow for the rapid diffusion of stuff across it. Any questions on that? Okay, our next type is simple cuboidal. This is what's very easy to see in your kidney slide. This will be primarily what you see. That's why I don't recommend looking for simple squamous there. You can, and I can help you find it, but it's easier to see simple cuboidal. Simple cuboidal cells, you can see the basement membrane here, and then all of our little cuboidal cells making this little tubule. And what simple cuboidal cells are good for is secretion and absorption. So what happens in the kidneys is we're going to filter the blood in our simple squamous layer and produce this fluid that now we're going to adjust. We're going to pull things out of it that we need, we're going to put things into it that we don't. So we can secrete and absorb very easily across this cuboidal layer. So that's going to be the main function of a simple cuboidal tissue, is that it helps with secretion and absorption. So you find it in your kidney tubules, and then in some of your small glands, it forms the secretory portion that is making up the part that's producing and releasing products in your glands. And then also on the surface of your ovaries. We'll talk about ovaries next semester. Questions on simple cuboidal? All right, simple columnar is next. And again, you can see your basement membrane down here, and we've got our basally located ovoid-shaped nuclei, and then it's facing the lumen out here. This is showing us what are these cells in the small intestine. And the small intestine has the super important job of finishing up nutrient digestion and absorbing all of your digested nutrients. So the small intestine has tons of microvilli all over it. And you have some slides that you can actually see them, which is pretty cool, because it can be hard to see things so small. So this is a simple columnar layer. And if you think about why would we want simple columnar as opposed to simple cuboidal if what we're doing is absorbing and secreting still? Well, these cells are continually exposed to digestive enzymes. So they're a little more sturdy if they're columnar than if they're cuboidal. So this is you'll find our columnar cells throughout the GI tract because they're a little more sturdy than our cuboidal cells. Another cell type that you can see here that you're going to have to identify under the scope is what's called a goblet cell. Goblet cells secrete mucus. So you find them all throughout epithelial tissues when we're lining mucus or making mucus membranes or lining all of our wet organs of our GI tract. And they look white because mucus doesn't pick up dye. So they're pretty easy to see. They're very distinct and they're very nicely circular shaped. So you have to identify those as well. So the function is really going to be in the small intestine absorbing nutrients, but they also are simple columnar epithelial tissues, secrete mucus because of the goblet cells. They also secrete digestive enzymes. That's going to be really important, and we'll talk more about those next semester. You have ciliated versions. This is not a ciliated version. This has microvilli, which is going to increase surface area. So that's a mistake a lot of people make. They think that microvilli move stuff. They do not. Microvilli increase surface area. Cilia move stuff. So we've got a ciliated version that will help in areas where we need to move mucus along. So like your respiratory tract. In your lower respiratory tract, we have what's called the mucus escalator. And it catches anything in your upper respiratory tract. It's a different kind of ciliated tissue we'll look at in a second. But when you get down to your lower respiratory tract, we'll have these simple columnar cells that have cilia on them. And if you inhale something like dust and it makes it past the mucous membranes in your nose and gets into your tubules, we need to get it out of there. So these cilia beat the mucus up. It's called the mucus escalator. And if we can get it back to your pharynx, then you can swallow it. And if it goes to your stomach with the hydrochloric acid, you can digest it. So if there was any kind of pathogen in there or anything in there that's dangerous to your body, you can digest it in your stomach, which is a pretty cool thing. We'll talk about that more next semester as well. This is what we'll find in your nasal cavity. Is what we call pseudostratified columnar epithelium. And it's pseudostratified because it looks stratified. It looks like there are many layers because the nuclei of these cells are all located at different places. But if you'll notice, this cell here, whose nucleus is up here, is still attached to the basement membrane. So every cell is still attached to the basement membrane. It just looks stratified because the nuclei are found at different distances from that basement membrane. So pseudo means fake. So this is the fake stratified columnar epithelium. The cells are still columnar, they're taller than they are wide, and these cells, we can see, have goblet cells as well. You have to have mucus in your respiratory tract, and these cells are the ones that have cilia. So you can find these in your trachea, you can find these in your nasal cavity. It's your larger bronchioles and things that contain the our simple, not pseudostratified version. But in your trachea and your nasal cavity, you can find our pseudostratified ciliated columnar epithelium. And again, they're going to secrete primarily mucus, and then their job is to pulse that mucus along so that it moves either back to your pharynx for swallowing or up to your pharynx for swallowing. We also have it very rarely found in the ducts of the male reproductive tract that carry sperm, but the non-ciliated type, and so it's pretty rare to find the non-ciliated type. The ciliated variety is found through the trachea. A lot of the upper respiratory tract has that as well, and its big job is going to be to move mucus. Yeah?
Goblet cells, are they they're in this one, the simple columnar, are they in the cuboidal as well?
In some portions of your respiratory tract, when you start moving down your bronchial tree, I wonder if I've got a bronchial tree anywhere for you to see. I don't. But when you start moving down your bronchial tree, the layers of the epithelium gets thinner and thinner. So you would have, it would go from columnar through cuboidal until we get all the way down to simple squamous. And so you can also find it in cuboidal. Basically, you find goblet cells anywhere you need mucus. So they've only been imaged in the last couple slides, but in like the lower portions, this is kind of a misnomer about this. In the lower portions of your bronchial tubes, you have simple cuboidal that would also have goblet cells. So they're just anywhere you need mucus. And in your mouth, it's stratified squamous epithelial tissue. You've got goblet cells there as well. So anything that's wet has a goblet cell.
So even simple squamous.
When you get down to the alveoli, we're no longer making goblet cells, or have goblet cells that are making mucus. They make stuff called surfactant, but we'll talk about that next semester. All right, next let's talk about our stratified epithelial tissues. So our first is the stratified squamous epithelial tissue, and there are two versions. There's one called keratinized that contains a waterproofing protein called keratin, and there's one that's non-keratinized. And they're stratified squamous, so they're stratified because they have many layers, and they're squamous because the top layer, or the apical surface, has squamous-shaped cells, but if you look, the cells on the basal surface look very different. They look like, sometimes you can't tell, but a lot of times they look cuboidal or something like that. So we're naming the tissue based on the shape at the apical surface, and this tissue is stratified because it has many layers. And this example is our stratified squamous epithelial that is lining the esophagus. And if you think about the esophagus, it's continuously exposed to abrasion as you swallow food. So the function of stratified squamous epithelial tissues is to protect everything underneath it by being able to resist abrasion. So our stratified squamous epithelial tissues are abrasion resistant. And in the esophagus, this would be an area where we still have goblet cells because it's got to be mucus. And in our esophagus, we have the non-keratinized version. So there are two versions of stratified squamous epithelia, non-keratinized, and these do not contain keratin. So we find these in your esophagus, in this part of your pharynx called your oropharynx. You find it in places like the vagina, anywhere that is going to be exposed to abrasion. So you're lining your mouth, lining part of your pharynx, lining your esophagus and your vagina. So anywhere that is exposed to abrasion has to have the stratified squamous, and if it's a wet surface, it's got to be the non-keratinized version. Because keratin is a waterproofing protein. So there's a keratinized version, and we'll say this contains keratin. And what keratin does is it's a waterproofing protein. So it helps to be able to, like in your skin, it helps to resist like rapid accumulation of fluids. Now it's not 100% perfect. We've all sat in a bath too long and become raisin fingers, but we're not absorbing the whole bath. We're also not losing all of our own moisture because keratin is helping us to retain it. So this is a waterproofing protein. And you find this, the keratinized version, in the epidermis of your skin. So it's dry, and it helps to, this is really going to help to make this skin the nice solid boundary between you and the external environment. So that you're not losing too much water, so that you're not taking on too much water. All right, our last type of epithelium that we're going to talk about is what's called transitional epithelium. This is only found in one place in the body. It's called transitional because the cells on the apical surface can transition in shape depending on level of stretch of the organ. The organ is your urinary bladder. So your ureters, which bring urine to your urinary bladder, and your urinary bladder both are lined with transitional epithelium. And so it's transitional because it transitions, the cells on the surface transition in shape. And when the bladder is empty, they tend to look cuboidal, but then as it fills, they stretch out and look more squamous. So we'll say this is only found in ureters and urinary bladder. And it's called transitional because our apical cells transition in shape depending on level of stretch. So when your bladder is empty, they look cuboidal. When your bladder is full, they look more squamous. We'll talk more about the bladder next semester as well. Any questions so far about our different types of epithelial tissue? Then the last thing we're going to talk about before break is glandular epithelium. And so glands are things that produce and release a secretion. And we've got two types of glands. Endocrine glands that produce a hormone and release it to the blood, and we'll talk about those starting next semester. It's the first system we talk about. So these produce a hormone and release it to the blood. Anytime you see the word endo, you can think within. So these are going to produce things within your body and release it within your body. The other type of gland we have is exocrine. Exo means without. So they produce a product and secrete it into a duct that dumps onto an epithelial surface. So endocrine glands we'll talk about next semester, produce hormones, release them in the blood. Exocrine glands, we're going to talk generally about them today, their shapes and their method of release. We're going to talk about specific exocrine glands when we get to the skin next week. So our exocrine glands produce a product and release it into a duct that dumps onto an epithelial surface. So one thing, like some things people classify endocrine glands as being ductless. Our exocrine glands all have ducts, which are just these little tubes that are going to pick up the secretion and bring it to an epithelial surface for release. So we'll talk mostly about most of our exocrine glands this semester. Next semester we have to talk about pancreatic glands, so when we get to the digestive system. As far as the way that we classify our exocrine glands, we can classify them based on two things, either structure or the way that they release their product. So exocrine glands, we can say, are classified based on structure or based on method of release. So just be aware, I'm not going to like ask you to name some compound tubular gland specifically. I just kind of want you to be aware of the ways that we classify them based on structure. So we call our glands simple if they've got one duct that's conducting the product away. Tubular if the secretory portion, which is producing the product, is about the same size and diameter as the duct. So this would be like simple tubular. The secretory portion is about the same diameter as the duct. It's tubular because of that same diameter. It's simple because we've just got one duct. You can have simple branched tubular where you've got several secretory portions dumping into one duct. You don't have to frantically write all this down because I'm not going to test you on this. This is just something to be aware of. So if you've got like branching secretory portions dumping into one duct and they're all about the same diameter, that would be simple branched tubular. Compound structure is when you have several duct parts. So this would be compound tubular because our secretory parts are as big as the duct parts, but then we've got several ducts that are branching to dump into this one duct on the epithelial surface. The second type we can classify based on the secretory structure is what's called alveolar. This is also called acinar. So we'll say based on structure, tubular is same diameter. Our secretory portion has the same diameter as the tube. The other type we have, your book is calling alveolar. I'm having spelling problems. Alveolar, some books call it acinar. This is going to be important when we look at the pancreas. This is why I bring it up, because a lot, I think maybe even in your lab manual it calls them acinar. So alveolar and acinar secretory portions are larger than the duct portion. So they tend to bulb out around the duct. And we can have simple alveolar again, where it's one secretory region dumping into one duct, and the secretory region is bigger than the duct. We can have simple branched, simple because it's one duct, but branched because we've got several secretory portions. We can have compound alveolar, where we've got several ducts, several secretory portions pouring into those ducts, and then dumping onto the epithelial surface. And then the most complex kind is where we've got compound tubular alveolar. So we've got both types of secretory portions. Some that are the same diameter as the duct, some that are enlarged relative to the duct, and lots of branches. I'm not going to make you name all of these things. I want you to be aware that these types of glands exist in the body. Why? Because if we cut, if you have to identify like a slide, our slides, they're just cutting through one section and you're looking at something. So if you were looking at this duct and you were to cut through it, what you would see would be a whole bunch of little, what look like individual tubules that are surrounding, or secretory portions surrounding ducts. But what you're doing is you're actually looking at the same gland. We've just cut through it so you can see two parts of it. So in the kidney tubules, when you look at that compound or your simple columnar epithelium, it looks like a bunch of these little individual things. But what it is this tube that's twisting back and forth, and we've just cut it in one spot. So just be aware of that, so that we can classify our exocrine glands based on their structure. Tubular or alveolar referring to the secretory structure, simple or compound referring to the duct structure. The other way we classify our exocrine glands is based on method of release. So we've got three methods of release. The merocrine method of release is when your secretory cells are releasing their product through exocytosis. So most of the sweat glands in your body are like this. So they produce sweat and they release it by exocytosis. So based on method of release, we would call them merocrine, and we would say that the secretory product is released through exocytosis. So you make your stuff, you package it into a vesicle, the vesicle fuses with the plasma membrane, and you dump it to the outside. So most of your sweat glands are a good example of that. You have two types of sweat glands, so not all of your sweat glands are that, but the ones all over your body, sweaty palms, are American sweat glands. They have a specific name we'll talk about next week. The second way that we can classify exocrine glands based on method of release is what's called holocrine glands. And holocrine glands are weird because they accumulate their product and then the whole cell ruptures, releasing that product and cell fragments into the duct. So holocrine are pretty, it's kind of gross. An example of a holocrine gland, all of your hair cells, you've got little tiny hairs everywhere. All of your hair cells have glands, holocrine glands near them called sebaceous glands. Sebum lubricates your hair, makes it soft and pliable. So it's this like oily, protein-rich substance that these cells accumulate and then the cells disintegrate. So bacteria are all over your skin. They love to eat that stuff up. So the major component of pimples is bacteria that you're killing off because they're going in, they're climbing down this hair, and they're gobbling up all this stuff that's being released from this sebaceous gland. So it's kind of weird. It's kind of a unique way for glands to release a product, but they accumulate the product, and then this whole cell disintegrates. So they produce and accumulate a product and then disintegrate. And when we talk about skin next week, we'll talk about all of the specific glands. This is the general way of classifying them based on method of release. Next week, when we talk about the skin, you have to start learning the specific names of the glands in the skin that are doing this. So they produce and accumulate a product and then disintegrate and release everything, and release product and cell fragments into the duct. There's not a picture for the last type based on method of release, but I'm going to tell you about it. They're called apocrine glands. And in apocrine glands, it's kind of like this weird hybrid between the two. So our apocrine glands accumulate a product on the apical surface, and then they pinch off from the apical surface. So it pinches off at the top. So if I were to draw a picture of it, it would be like they bud off, and then like if this is the cell, it's going to pinch off. So it forms its product at the apical surface, and then it pinches off and is released into the lumen. So they will say, produce the product on apical surface that pinches off into the duct. Mammary glands work this way. So they produce milk proteins at the apical surface, and then pinch off or bud off into the lumen to release that out to the duct, the lactiferous duct that's going to carry it away to the surface of the nipple.